This question is about photosynthesis. Complete the word equation for photosynthesis. So this is a nice fact recall. Carbon dioxide plus water, arrow, glucose plus oxygen. Describe how energy for the photosynthesis reaction is gained by plants. That is because light is absorbed by chlorophyll. Students investigated the effect of temperature on the rate of photosynthesis. The students shone light from a lamp onto pondweed and measured the volume of oxygen produced per hour. Table 1 shows the results. The different temperatures, our independent variable there, temperature in degrees C, 20 to 45 degrees C, and our rate of photosynthesis as the volume of oxygen produced per hour. Calculate the mean value of x. We need to add 18.5 plus 19.3 plus 19.5. That comes to 57.3, and we did 3 tests, so we need to divide that by 3. That gives us 19.1, and we just need to check how many decimal places we should do our answer in. And if we have a look at the whole mean column, all the means have been calculated to 1 decimal place. So that's how we know we've got to write 19.1 there. The students identified one anomalous result in Table 1. Draw a ring around the anomalous result in Table 1. So have a look at all the rows. Is there any value that stands out as being quite different from the rest in the row and very different from the mean as well? The one that stands out is at 45 degrees C. You can see that test 2 is 14.2. And that's really different to the other 2 sets of results and also to the mean. Suggest one possible cause of the anomalous result. Maybe there was already air or oxygen in the measuring cylinder or gas syringe. We only need to write one down, but there could be other examples like the pondweed had not acclimatized. That means hadn't had time to get used to that temperature. Maybe the temperature changed from what we were setting out to investigate. Maybe the lamp was moved. How did the students deal with the anomalous result? You need to check the data, but in this case they have discarded it, which is what we'd want to do. We'd want to discard it and don't use it to calculate the mean, but you've always got to check whether they did that, because there might be an exam question where they haven't actually discarded it. Pay attention to the table. Give one factor the students should have kept constant in this investigation. Well, we know we're changing the temperature. One to keep constant would be light intensity. Why did the rate of photosynthesis decrease from 35 degrees C to 45 degrees C? What we need to remember in biology is that all biological reactions rely on enzymes. So when you're thinking about respiration and photosynthesis, for example, they're always going to be reliant on enzymes. So we kind of know when the temperature starts to increase, we have got a lower volume of oxygen being produced. So maybe the enzymes are starting to denature, and that would mean they're losing their specific complementary shape of their active site. So these enzymes that are needed for photosynthesis to take place. Complete figure 1 using the data from table 1. You should label the y-axis, use a suitable scale for the y-axis, plot the mean from table 1 for the temperatures from 25 degrees C to 45 degrees C. Draw a line of best fit. I'm just writing the values down on the side here so I can easily see them when I'm plotting it on this tablet. But obviously in the paper you can just flick back and have a look at the data. We've got to label the y-axis and the volume of oxygen collected, so the rate of photosynthesis centimetres cubed per hour, varies from 2.1 to 44 being the highest. So I think it's perfectly reasonable that we could keep this y-axis really simple. Every 1 square would be 1 centimetre cubed per hour. 5, 10, 15, 20, 25, 30, 35, 40, 45. Brilliant. And then we can plot our points. And as you can see, our line of best fit looks very much like a curve, so that's what we need to do. And there's our curve of best fit. Diffusion is an important process in animals and plants. What is meant by the term diffusion? Movement of molecules or particles from an area of higher concentration to an area of lower concentration down the concentration gradient. Figure 2 shows part of a leaf. Molecules of carbon dioxide diffuse from the air into the mesophyll cells. Which two changes will increase the rate at which carbon dioxide diffuses into the mesophyll cells? Will it be a decreased number of chloroplasts in the cells? No. Decreased surface area? No. Increased carbon dioxide concentration in the air? Yes, because we'll have a lovely concentration gradient for the CO2 to move down. Increased number of stomata that are open? Yes. The more that are open, the more the carbon dioxide can diffuse in. Diffusion also happens in the human lungs. Figure 3 shows the human breathing system. Explain how the human lungs are adapted for efficient exchange of gases by diffusion. So I'm just going to annotate this diagram here so I can remember to cover as many bases as possible in this 6 marker. First of all, we can see that person's left lung. They've drawn out the alveoli, and we can see that we've got lots of alveoli there. We can see if we look at one alveolus, then the capillary, notice the capillary wall, if you remember, is actually 1 cell thick, and it's very close to the wall of the alveolus as well. The alveolar wall is actually 1 cell thick as well, so that's going to help with a short diffusion distance. Also, there are going to be loads of capillaries as well, and breathing in brings air into the alveoli, which gives us the fresh supply of oxygen, and breathing out is going to remove the carbon dioxide. Breathing in helps to maintain the concentration gradient from the alveoli into the blood. Breathing out helps to maintain the concentration gradient for the carbon dioxide to diffuse out of the blood and into the alveolar and then out completely. So let's write as many points as we can fit on these lines given. So the many alveoli provide a large surface area for the diffusion of gases. Nice key terms there. The capillaries are only 1 cell thick and are very close to the alveoli. The alveoli are also only 1 cell thick, so the diffusion distance between the blood and the alveoli is very short. This means oxygen can diffuse quickly from the alveolus into the blood, and the carbon dioxide can diffuse quickly from the blood and into the alveolus to be breathed out. The breathing in and out ensures that oxygen is brought into the lungs and the alveoli, maintaining a concentration gradient so that oxygen can always diffuse into the blood. Breathing removes carbon dioxide. The actual movement of the blood in the capillaries maintains a concentration gradient. So if you think about it, as the oxygen diffuses into the blood, and it will bind with the hemoglobin, won't it? And as the blood moves along, the next bit of blood is quite low in oxygen concentration, so oxygen can move from the alveolus into the blood and so on and so on. So that helps to maintain a concentration gradient. And also that concentration gradient is further helped by there being many capillaries providing a good blood supply. And that's all we can fit. I think we've covered most of the points there. Figure 4 shows a root hair cell. Name the process by which water molecules enter the root hair cell. We know that water molecules will only move by osmosis, from a high water concentration to a low water concentration through a partially permeable membrane. Nitrate ions need a different method of transport into the root hair cell. Explain how the nitrate ions in figure 4 are transported into the root hair cell. Use information from figure 4 in your answer. So if we have a look at figure 4, we can see the nitrate ions are represented by these crosses. And if you have a look inside the root hair cell, there's a much higher concentration of the nitrate ions inside the root hair cell compared to outside. If the plant's trying to get the nitrate ions into the cell, then those nitrate ions are going to have to move from a low concentration to a high concentration. So that means against the concentration gradient, which we know is active transport, and it's active because it needs energy to be able to do that. So the name of the process is active transport. Energy is needed to transport nitrate ions from a lower concentration in the soil to a higher concentration in the root cell against the concentration gradient. Figure 5 shows 3 types of cells. We have a bacterial cell, a liver cell, and a mesophyll cell. Give 2 similarities between the prokaryotic cell and the eukaryotic cells. So our bacterial cell is the only prokaryotic cell there. The liver cell is eukaryotic and it's an animal cell. And the mesophyll cell is a plant cell and it's also eukaryotic. So we need to look at what is similar about all 3 of those cells. And that would be the cytoplasm and the cell membrane. You could also have put ribosomes or DNA, genetic material. Give 3 differences between the prokaryotic cell and the eukaryotic cells in figure 5. The question is asking you 3 differences between the prokaryotic cell and the eukaryotic cells. So you can't use cell wall in your answer because the eukaryotic animal cell doesn't have a cell wall. So it needs to be differences between the prokaryotic cell and both eukaryotic cells together. For example, eukaryotic cells have mitochondria and prokaryotic cells don't. Both the animal and the plant cell have the mitochondria. Should have gone with this one first, really. Eukaryotic cells have a nucleus. Yeah, both the animal and the plant eukaryotic cell, of course, they have a nucleus. Prokaryotic cells have plasmids and eukaryotic cells do not. And I just want to draw attention to a little bit of misleading bit in the diagram. The plasmids you can see quite clearly in the bacterial cell. They're drawn in a similar way to how they've drawn the chloroplast in the mesophyll cell, but they are different. So that's why you need to rely on your knowledge as well as looking at diagrams. Calculate the ratio of the size of the bacterial cell to the size of the mesophyll cell. So the bacterial cell is 1 micrometer, which we can get off the diagram. The mesophyll cell is 0.05 millimeters. So we need to make those to the same unit so that we can get a valid ratio. And we know that there are 1,000 micrometers in 1 millimeter. So if we times 0.05 millimeters by 1,000, that will give us 50 micrometers. So if the bacterial cell is 1 micrometer and the mesophyll cell is 50 micrometers, then we're going to have a ratio of 1 to 50. Name the type of cell division that produces genetically identical body cells for growth and repair. Mitosis. Figure 6 shows a cell cycle. So we've got stage 1, stage 2, stage 3. What percentage of the time for one cell cycle is represented by stage 2 and stage 3 together? So if you add up the segments in stage 2 and 3, that comes to 7, and in total there are 20 segments in that whole circle. So we just do 7 divided by 20, that gives 35%, and that's our answer. Describe what happens during each stage of the cell cycle. DNA replicates, so that means the chromosomes that are made of DNA are going to replicate as well, and the number of mitochondria and ribosomes also replicate while the cytoplasm is growing. Stage 2, one set of chromosomes is pulled to each end of the cell. So we're getting ready for a division. And in that 3rd stage, the cytoplasm and the cell membrane divides to form 2 cells. And that is called cytokinesis. Lipases break down lipids. Which 2 products are formed when lipids are broken down? Fatty acids and glycerol. One model used to explain enzyme action is the lock and key theory. Figure 7 shows a model of the theory. So I'm just going to have a look at this diagram here. We can see that this is the active site of the enzyme. We can see that the enzyme and the substrate are complementary shapes. We can see in this next section of the lock and key theory that the enzyme and substrate will bind to each other, form an enzyme substrate complex, and then a reaction will happen. And in this example, it looks like the bonds are broken in the substrate and then the enzyme's active site releases the products. Let's answer the question then. Explain the lock and key theory of enzyme action. Use the information from figure 7 in your answer. So we just have to put that into words and make sure we've got our keywords being used. The active site of the enzyme will collide with and bind to the substrate with a complementary shape. The bonds are broken in the substrate, or it's broken down, into products that no longer fit the enzyme's active site, and the enzyme can be used again. There are many different types of lipases in the human body. Why does each different type of lipase act on only one specific type of lipid molecule? Each active site has a specific shape only complementary to one type of lipid molecule. Students investigated the presence of starch and glucose in the leaves of geranium plants. This is the method used. Place 2 identical geranium plants on a bench near a sunny window for 2 days. After 2 days, leave 1 plant near the window for 2 more days. Place 1 plant in a cupboard with no light for 2 more days. Remove 1 leaf from each plant, crush each leaf to extract the liquid from the cells, and test the liquid from each leaf for glucose and starch. Describe how the students would find out if the liquid from the leaf contained glucose. So they would add Benedict's solution to the liquid and heat in a water bath for a few minutes. I've underlined heat because you don't get a mark for just saying put it in a water bath. You have to tell the examiner that you understand that you have to add the Benedict solution first, and then you've got to heat them both together. If glucose is present, the Benedict solution will change from a blue to either a yellow, green, orange, brick red colour. You get a mark for any of those colours because it depends on the concentration. Describe how the students would find out if the liquid from the leaf contained starch. Add iodine solution to the liquid, and if starch is present, the liquid will change to a blue-black colour. Table 2 shows the students' results. So we have got the leaf from the plant kept in the light for 4 days, and the leaf from the plant kept in the light for 2 days, and then no light for 2 days. And we've got a strong positive glucose, positive starch for the one in the light, weak positive glucose for the one in the dark for 2 days, and negative starch. So explain why the leaf in the light for 4 days contained both glucose and starch. Well, glucose is being made by photosynthesis, and some of that excess glucose is stored as starch. Explain why the leaf in a cupboard with no light for 2 days did contain glucose but did not contain starch. I think it's worth remembering that that's a weak positive for glucose as well. There is no light available for photosynthesis, so glucose can't be made. The only way for the plant to get glucose is by converting the starch into glucose. So that means there will be some glucose there, but just not a lot because photosynthesis isn't happening. The plant needs this glucose so that it can carry out respiration and release energy needed for a variety of processes, like active transport, for example. Suggest one way the students could develop the investigation to find out more about glucose and starch production in plants. You could test other species of plant. Maybe you could measure the concentration of glucose and starch. Many plants have evolved defense mechanisms. Figure 8 shows part of a Gauss plant and part of a deadly nightshade plant. The Gauss plant has evolved to have sharp thorns. What type of defense response are thorns? Well, those are mechanical. I prefer physical because that ties in more with A level as well, but either of those are correct. How do thorns defend the Gauss plant? It's going to prevent animals, mostly herbivores, are going to be eating them or damaging it. The deadly nightshade plant has poisonous berries. What type of defense response are poisonous berries? Chemical. A scientist noticed that in one area, the Gauss plants had yellow leaves and had stunted growth. One reason for yellow leaves and stunted growth is a deficiency of nitrate ions in the soil. Explain 2 other possible reasons for yellow leaves and stunted growth. Do not refer to nitrate ions in your answer because they've already mentioned that in the question. So it's very nice of them to remind you that. It could be infected with a pathogen such as tobacco mosaic virus. And if you remember, the tobacco mosaic virus causes discoloration of leaves. So that would mean less chlorophyll will be present, which means less light will be absorbed and less photosynthesis can take place. And that will mean less glucose will be made, so not enough energy for growth. It could also be due to a lack of magnesium ions, and magnesium ions are important for making chlorophyll. So if there is a lack of chlorophyll, there will be a lack of photosynthesis and glucose being produced. Without glucose, the plant can't carry out respiration, so there won't be any energy for growth, such as building big molecules from smaller ones. The Gauss plant has nodules on its roots. The nodules are part of the living root tissue. Bacteria which convert nitrogen gas into soluble nitrate ions live in the nodule tissue. Figure 9 shows the nodules on the roots. Suggests how the nodules benefit the bacteria. It is an application question and they are only asking you to suggest. So our best suggestion would be that the bacteria could easily obtain the glucose because it says that they live in these nodules and that's part of the living root tissue. So the bacteria can get some glucose from the plant. And of course, the bacteria need the glucose for respiration. Explain how the nodules benefit the Gauss plant. The information to help you with that is at the top of the question, but I think quite tricky as well. You have to remember that amino acids have nitrogen in them. So if the Gauss plant obtains nitrate ions, it will be able to make plant amino acids and plant proteins. For many years, drugs have been extracted from plants. Which plant material was chewed as a painkiller? So this was a long time ago, and that would definitely not be foxglove leaves. Please don't do it with those. Foxglove leaves are poisonous. It would be the willow bark. Data from the Million Women survey in the UK was collected for over 15 years. So this looks like a huge study. Scientists analysed the data to study the effect of consuming alcohol on liver disease. The scientists included 400,000 women who regularly consumed alcohol, 400,000 women who did not consume alcohol, and excluded women who already had liver disease. Age and gender were 2 factors controlled in this analysis. Many other factors were also controlled, suggest 2 other factors which the scientists would have controlled. We need to make sure that these women have very similar diets. Fatty diets can actually lead to liver disease. We need to make sure there isn't already a family history of liver disease because that would show some kind of genetic predisposition to it, and we want to look at the link of alcohol to the liver. The data was analysed for women who drank alcohol with meals, women who drank alcohol not with meals, and women who did not drink alcohol. During the survey, approximately 1,500 women developed a liver disease called cirrhosis of the liver. Scientists calculated the relative risk of developing cirrhosis of the liver for each group who consumed alcohol. A relative risk of 1 means there was no statistical difference between the groups who did consume alcohol and the group who did not consume alcohol. Figure 10 shows a summary of the results. We've got our graph here with our key. A woman drinks 150 grams of alcohol per week, not with meals. The woman decides to change to drinking 150 grams of alcohol per week with meals. Calculate the percentage decrease in the relative risk of developing cirrhosis of the liver for this woman. So we need to go to the 2 lines and go to 150 grams per week on the graph. Take it up. So that top line there is the relative risk of developing cirrhosis of the liver is 2.55 because it's just slightly over that 2.5 line, which is a bit annoying, but it is 2.55. It's in the middle there. And if you go to consumed alcohol with meals at 150, that's bang on 1.6. We need to do the change in the relative risk. That's 2.55 minus 1.6. which gives us 0.95, And then we need to work out percentage decrease. So you have to divide the change over the original value, which was 2.55. Times that by 100 to make it into a percent. And we've got 37%.2 something or other. It doesn't tell us how many decimal places we should do that to. And the mark scheme is very happy for that to be a whole number there, or for you to have decimal places in it, either way. 1 glass of wine contains 12 grams of alcohol. A different woman drinks 2 glasses of wine each day with her meals. Calculate the relative risk of developing cirrhosis of the liver for this woman. So if she's drinking 12 grams of alcohol in 2 glasses, that's 24 grams, so that's per day. Then we need to times that by 7 days to work out how many grams of alcohol she's consuming in a week. It says with her meals, so we need to find 168 on the x-axis. And you can see that between 150 and 200, I was just trying to work out how many grams are per one little square. And that is 50, so it's 5. 155, 160, 165. Rather awkwardly, then we have to go a little bit between 165 and 170. Take that line up until it hits the line consumed with meals. Then draw a line across to the y-axis, and that gives us 1.8 risk of developing cirrhosis of the liver. And that's what we need to write down there. Figure 10 is repeated below, so same graph. Consuming alcohol with meals instead of not with meals decreases the relative risk of developing cirrhosis of the liver. Give 2 other conclusions about the relative risk of developing cirrhosis of the liver related to alcohol consumption. The thing that strikes me is that if you increase the alcohol consumption from 25 grams to 215 grams, which is where the consumed alcohol not with meals starts, 25 grams, and then it finishes at 215 grams, there is an increase from a risk of 1.1 to 4.5. That's an increase of 3.4 relative risk of developing the cirrhosis of the liver. The other thing to note is that, can you see the curve with consumed alcohol with meals? If the person consumes less than 50 grams per week of alcohol, then there's actually no increase in the relative risk. I'm sure there are more things in the graph to pick out, but those are the most obvious things I can see. Suggest 2 reasons why the data is considered to be valid. The information right at the beginning, it tells you there are 400,000 women drinking this, there are 400,000 women doing this. It was a large number of people used in the survey. And also the data was collected for 15 years, so that's a long time. Suggest 1 aspect of the survey which might reduce validity. Well, people could underestimate or overestimate their alcohol consumption, and they could even lie. Cirrhosis of the liver leads to liver failure. Describe the effects of liver failure on the human body. We talk about the liver in paper 1 material, and we also talk a little bit about the liver and different functions in paper 2. So my answer here is based around stuff you've learnt in paper 1. So the first thing I would go with would be, if the liver can't make bile, then the pH of the small intestine may not be neutralised, meaning the enzymes in this area might become denatured. Lipids won't be emulsified, so their surface area will not be increased, meaning that lipases may not work effectively, resulting in lipids not being absorbed properly, so the individual could lose weight. Lactic acid is usually converted into glucose by the liver, but if the liver is not working, then the lactic acid will build up and it's toxic. They do accept some of the paper 2 answers on there if you happen to have written them. So the liver not being able to convert excess amino acids into urea, you learn that when you do about the kidney, but you didn't need to put it here for the answer, but if you did, then you would have got some marks for it. And the other thing that you don't learn until paper 2 is about making glycogen stores with excess glucose and insulin and things like that. So again, if you wrote about that here, they wouldn't deny you the marks, but I didn't put it here because I felt I wanted to stick with paper 1 stuff that was fresh in your head, and you can get the marks for that anyway. Monoclonal antibodies are usually made using mouse lymphocytes. Notice that they have put monoclonal antibodies as an abbreviation, so you're free to use that in your answer if you wish to. Candida albicans infection produces serious symptoms in patients with a poor immune system. Recently, scientists have produced monoclonal antibodies to Candida albicans using human lymphocytes produced naturally after an infection. Candida albicans lives in the throat of infected patients. A sample is taken from the throat of the patient with suspected Candida albicans infection. The sample is transferred onto a microscope slide. Describe how the monoclonal antibodies and a fluorescent dye could be used to see any Candida albicans pathogens on the slide. You could bind a fluorescent dye to the monoclonal antibodies, put these monoclonal antibodies onto a slide with the sample taken from the throat. So if Candida albicans is present, the monoclonal antibodies will bind to the Candida albicans and show up under a microscope or a UV lamp. In a laboratory, the human lymphocyte monoclonal antibodies were injected into animals infected with Candida albicans. The monoclonal antibodies caused increased phagocytosis of the Candida albicans pathogens. Doctors intend to start a trial to give the monoclonal antibodies to patients severely ill with Candida albicans. Explain how increased phagocytosis of the Candida albicans pathogen will help the patient. More Candida albicans will be engulfed and destroyed by phagocytes, and therefore there will be less damage to the cells by this Candida albicans. It has been shown that this monoclonal antibody treatment is effective in the laboratory using both infected tissue culture cells and infected live animals. The monoclonal antibody treatment from Candida albicans is now ready for clinical trials on people. Describe how the clinical trials should be carried out. So first, the treatment should be given to healthy volunteers at a low dose to test that it is safe and whether there are any side effects. A placebo will also be used in some healthy volunteers so that the results can be compared. Then some patients with the disease will be tested on to try to get the correct dose, to check for any side effects and to test for the efficacy of the drug. So that means how effective the drug is. A double-blind trial is necessary so that neither the patients or doctors know which patients have received the drug and which have received the placebo, therefore ruling out any bias, seeing patterns in the data that aren't there, or just generally misinterpreting the results. The drug needs to have been tested on a large number of individuals over a long period of time. Last one. Scientists have also used human lymphocytes to make monoclonal antibodies to other pathogens and to some types of cancer cells. Suggest one reason why these new monoclonal antibodies have been more successful in treating diseases in humans than monoclonal antibodies made using mice. Humans are less likely to reject monoclonal antibodies made by human lymphocytes, or mouse monoclonal antibodies are more likely to be rejected by humans. And that is it. Well done for getting all the way through that paper. All the best in your exams, and I'll see you in the next video.